species. We need to know what a species is. And unfortunately, it's not an easy thing to define. And there isn't one definition out there that works for every living organism on planet Earth. Mainly because of the diversity that we see in terms of reproduction, in terms of appearances, even within species. It provides some complications for actually trying to define them. One of the main definitions that we use, though, is the biological species concept, and that is something that is based off of reproductive isolation. And so in order for two individuals to be considered the same species, they have to be able to interbreed with each other successfully, and they have to be able to produce viable fertile offspring. Okay? So we walked ourselves through the steps of sexual reproduction in the last class, where we brought our gametes together through the process of mating. They fused in the process of fertilization to make a zygote. Okay? That zygote then developed over time, okay? And then became a reproductive adult. And all four of those steps have to be met in order for two individuals to be considered the same species. And we said that you can stop any one of those steps. And if you do, then those parents are different species. You can stop mating, you can stop fertilization, you can stop the process of development, or you can prevent that offspring from being able to reproduce. We got through all of the pre-zygotic mechanisms. Those are the ones that happen before our zygote forms. Either they stop mating or they stop fertilization. What we were working in our last lecture was dealing with the post-zygotic isolation mechanisms, which means that mating has been successful between these two parents, and fertilization occurred. We made a zygote, a hybrid zygote, but then something is happening now beyond that point that is preventing that last criteria that the offspring is viable and fertile from happening. And so seeking compatibility was the first one we talked about, where you get your hybrid zygote. You may even get the embryo, which is a multicellular stage just beyond the zygote. It's not a mature adult yet, but you don't actually get a viable seed. The seed's not able to form. Right? And one of the reasons for that might be because you have a different set of directions, different parental genomes coming from the father and mother that are telling this organism to function, to grow, develop in completely different ways, and you end up with something that's completely non-functional. So we don't actually get a seed. And because their offspring is not viable, they are therefore considered, the parents are considered different species. Then we talked about hybrid inviability, which means that you get to the seed point. You may actually even get a seedling beyond this point, but it's not going to make it to adulthood because, again, in those cases, what's happening here is you've got parents that are in different required habitats or different types of environments they're adapted for. Their offspring is some hybrid thereof, okay, so some combination thereof, and that habitat doesn't exist. All you have are the habitats the parents are in. You don't have something in between, and therefore there's no habitat for them to be able to survive in. So that would be an inviable hybrid. They can't live in that location. And so that would also be post-zygotic isolation mechanisms. And in some cases, the hybrids just don't survive very long. They're very frail, again, because they've got different parents that are adapted for different things. Then we talked about hybrid floral isolation, where you have the parents adapted for different pollinators. And so you have a flower that is structured for bee pollination, one parent, for example, and you have another one that has a flower that's constructed or structured for pollination by hummingbird. Remember, they sense the world in different ways. They're attracted to different things. One needs to land on the flower, one doesn't. Right? Mouth parts are different. All of those things are important when it comes to a flower as far as what they're going to be able to pollinate. Now they have an offspring that's going to produce a flower that's kind of some combination between these two, which may not be something that either a hummingbird or a bee is going to be able to access. Right? Think about it in terms of floral isolation, which we talked about before, the pre-zygotic mechanism, where just the way the flower is shaped, right? Or the slit of the stigma in the milkweeds, right? Or the way the pollinia is shaped, it's like a key and a lock. If things are not structured correctly, there's just no way the pollinator can actually access that. It's just not going to happen. So it is similar to that process, as we have floral isolation, a structural issue with the flower here, but it's hybrid floral isolation because the parents have actually made it past those first two steps, mating and fertilization. It's the offspring that's having the problem. But the offspring have to be able to reproduce in order for these parents to be the same species, and if there's no effective pollinator, they can't do that. Okay, so that would be a little bit different than the pre-zygotic version of floral isolation. Now, we get left off talking about hybrid sterility, which is a situation where everything gets done properly. We get mating, we get fertilization, we get a viable offspring, they're able to grow to adulthood, they're healthy, they are mature, but they're sterile. They're unable to produce functional gametes, and therefore they can't mate with each other. The example I gave you was actually an animal example first, where you cross a horse with a donkey. You get a mule. Mules are perfectly capable of growing to adulthood. They're strong, they can live long, normal lives, but they can't produce functional gametes. And you can't have a mule mate with another mule, it doesn't work, they're sterile. Same thing applies here. If you cross this plant here, the brassica, with the raphanus, you get something that can grow. It has root systems, it can acquire nutrients, it can get water, it can photosynthesize, but it can't reproduce. And so that would be, again, violating one of the conditions of the biological species concept, that the offspring has to be fertile, has to be reproductive. Okay? Now, number 8, the new one that we're going to talk about today, is one of the more complicated ones, because everything actually happens. And all 4 of the steps of sexual reproduction get completed between these 2 parents. They mate, fertilization occurs, their offspring is born, the zygote develops and is able to develop to maturity, and that offspring is actually capable of reproducing. You might say, well, that doesn't make any sense. Then why would this then be considered reproductive isolation if they're able to do everything they're supposed to be able to do in order to match the biological species concept? It has to do with the generations that occur beyond this. Okay? So when you have your 2 parents here, we'll say parent 1, parent 2, they've crossed, they've mated, they've produced an offspring. That first generation is oftentimes referred to as the F1 generation. They are perfectly normal. They can reproduce, they can mature, they can develop, everything is fine with them. They can even mate back with the parents, or they could even mate with another F1 individual in that generation, perfectly fine. Okay. What is wrong is when you get down here into that next generation, what we refer to as the F2 generation. Then you start to see chromosomal abnormalities, okay? DNA combinations that are non-functional. Say starts to happen, and your fitness is lower in these later generations, F2, F3, and so forth. So you don't get a lot of long-term mixing in the gene pools beyond this point. And if you can't go really beyond this point and you have complications in those next generations, in some ways it is reproductive isolation. Right? So we still consider that a type of reproductive isolation. A good example in animals, because I know a lot of you are more familiar with animals, is fruit flies. This is what happens in a lot of different species of fruit flies when they cross with each other. That first generation you do it can do just fine, the next one is frail and is usually not going to be able to be reproductive. All right, well, this is kind of a table to help you remember what stages are actually being prevented in the sexual reproduction process for each one of these different types of reproductive isolation mechanisms. So remember, you have pre-mating, which is anything that happens to prevent the process, that first step, that mating process. You have post-mating but pre-zygotic, meaning you get through that first step, mating, but fertilization doesn't happen. So you still don't get your zygote. Both of these 2 categories right here are classified as pre-zygotic mechanisms because none of those the zygote actually forms. Okay? And then you have your post-zygotic, meaning that you get to the zygote, but something happens beyond that, either in the development or in the reproductive adult, with the adult being able to reproduce. So I've kind of broken them down into which categories they are. One of the things I want you to know for the test is be able to recognize examples of these. If I give you an example scenario to say, this is the problem that we're having when these 2 individuals reproduce. Which one of the reproductive isolation mechanisms is at play here? Right? Or I might ask you whether or not the zygote forms or doesn't. Right? Or what step in the process is being prevented for each one of these different examples. Okay? Each one of these different type of reproductive isolation mechanisms. And the nice thing about the post-zygotic ones is all of them start with hybrid with the exception of this one. So that's probably the toughest one to remember because it doesn't start with hybrid. All the rest of them, you know that you've made it to the zygote, right? Because it tells you that the hybrid was formed and then something happened after that. But that seeding compatibility is a little bit less obvious that it's post-zygotic, that the zygote actually does form. Some species can utilize more than one method of this. So for example, in animals, you have some that use something called ecological, right, or habitat reproductive isolation, where they don't live in the same habitats, therefore they never encounter each other to mate. But if you were to unnaturally bring them together, like for example a lion and a tiger, they could mate, fertilization would occur, you get a zygote, and you can get an offspring that develops. They can't, that offspring though can't reproduce, right? So factors are sterile. So sometimes you can incorporate or utilize more than one type of reproductive isolation mechanism to prevent that hybridization between the different species. Okay, well, this is all well and good. Right? We really should have a good idea now of what the biological species concept is, how it works, how we can use it to define different species. But now we're going to talk about why this works well for animals, but it doesn't really work necessarily very well for plants. It is pretty easy to test. All you have to do is you have to go out there and you have to find the two individuals, right, the two different groups that you want to see if they're the same species or not, and bring them together and see if they can interbreed and produce viable fertile offspring. They're easy to test, as long as they're living. Right? You can't do this with extinct species. This concept here doesn't work at all for extinct species. You can't do it for extinct. But as long as it's a living species, right, you can test this. But it doesn't still work well for plants. And here are multiple reasons why it doesn't. The first is, when you test this, you are bringing together two individuals and seeing if they can successfully interbreed. But when you do that with plants, you bring two individuals together, sometimes it works and sometimes it doesn't. You could bring the same two individuals together, and the first time they try and breed, it doesn't work. Second time it does. Third time it does. Fourth time it doesn't. Okay? It's not 100% one way or the other. Success of matings between individuals of different groups and plants varies anywhere from 0 to 100%. So it's not black or white here, in the sense that yes, they can interbreed successfully, or no, they can't. It's sometimes they can, sometimes they can't. So does that mean that when they can, they're the same species, but when they can't, they're different? That wouldn't work. We've got to have some sort of consistent definition. It has to be something that applies to it at all times. But it doesn't work that way when it comes to the breeding and interbreeding between different plants. The second problem is many plants can hybridize, and they do it frequently. We see it all the time in oaks. That's why I hate oak trees. Can't stand them. Absolutely hate them. They're really hard to identify as it is, and then the fact that they hybridize and they tend to show mixed characteristics makes it even more complicated. So there's some species that are perfectly capable of doing this. And if you were to define them by a different definition, in terms of what species they are, like for example their morphology, how they look, and we'd say these are different species, you would find that they'd be able to interbreed all the time. So that doesn't work real well if they can hybridize successfully. Third, a lot of plants can self-fertilize. They're mating with themselves. They're not interbreeding with anybody. In order to test this, you gotta be able to bring two individuals together and interbreed, but if they are mating with themselves, where's the other half of that pair? Right? There's nobody else they're interbreeding with, so by this definition, something that self-fertilizes itself would be its own species, because it's not interbreeding with anybody else. That's not the case, but that's what plants do sometimes. And in some ways, number 4 sort of applies in the same way. A lot of plants reproduce primarily through asexual reproduction. This concept only works for sexually reproducing species because the individuals have to interbreed. You have to have those 4 steps: mating, fertilization, development, reproductive adult. Okay? All of those stages have to be met in order for you to be able to apply this concept. If they don't, then automatically, by this definition, the species are considered different species. We'll take bacteria as an example. Bacteria can only reproduce asexually. That's all they can do. You cannot use this species concept for bacteria because they are never ever going to interbreed with each other no matter how many times you bring 2 individuals together. They just don't do that. So therefore, we would say every single cell is a different species, and that's not true. Well, it works that same way for plants. If they don't interbreed, if they reproduce asexually, then we can't define them as being their own species. Okay? But every individual would be its own species in that case. So it doesn't work for asexual reproduction. So let's talk about some of the different ways that plants reproduce asexually. There's a lot of different ways to do this. Okay? Asexual reproduction is basically any reproduction that occurs that makes a new individual that does not involve an egg and a sperm coming together. That's all it is. So there can be a lot of different things. One example would be something called stolons. Stolons are essentially horizontal stems that run above ground in different directions away from the original plant. Okay? You'll see them a lot in types of grasses. It's something that strawberries do. So if you watch your strawberry plant before, if you plant one plant By the end of the season, it starts to send out these lines, these runners, and like with lines going away from it. Anywhere that a node, which is where a leaf attaches, anywhere where a node touches the ground, it can actually form a new little plantlet. It'll send out new little roots, it'll bring up new little leaves, and you could actually cut the line between these two little plantlets right here, and this one would be able to live independently. It has its own root system to get water nutrients, it's got its own leaves to do photosynthesis compared to this one over here, but they are genetic clones of each other. They are genetically identical. That's one of the consequences of asexual reproduction. Everybody's genetically the same that gets produced by these processes. Another possible method of asexual reproduction that we see frequently in plants, similar but not exactly the same, is something that's referred to as rhizomes. Rhizomes are also horizontal stems. The difference is rhizomes are under the ground and stolons are on top. Right, so above ground, stolons you'll see, right, without having to dig up the plant, you can actually see those on top of the soil. But these, you're gonna have to dig down and find them. But they're still stems. People sometimes refer to these as false roots. Just go ahead and get that definition out of your mind because they are not roots. Anatomically, they are not roots, they are stems. You'll see the roots are coming off of them, right? So those are the roots. This is actually a stem. But it works the same way that a stolon does. Anywhere there's a node, you can get a new set of roots and a new little plantlet that forms at that spot. And again, once it's developed, you can cut that line and have them live independent of each other. This is one of the things that some of the ferns do. We've got a very aggressive fern in our greenhouse that does this. It spreads all the time, drives us crazy. This is what my lawn does. This is why I hate the lawn that I have, because it gets in my garden and I have to dig down to actually get these runners out. And it breaks very easily. So, and if you leave just a piece in there, it will still come back. So they're very effective at doing this. This is really common in grasses. You kind of have two different types of grasses out there. You have rhizomatous grasses, the ones that do this, and you have clump grasses, the ones that form into little bunches. All right, we also have bulbs as a means of asexual reproduction. A bulb is essentially a bud. It's an underground bud that has these thickened fleshy scales, think layers. That's why an onion has layers. Those are the scales that are wrapped around the bud itself. That's not a seed, right? This is a vegetative propagule, it's something different. And it spreads by division of bulbs. So you can see, here's the main bulb right here. It starts to form these little lateral bulbs right here. Think a garlic bulb or a piece of garlic. If you go to the grocery store, you'll find as you dig down underneath that paper, there's these separate little fragments you can peel apart. Those are the individual lateral buds that you can pull off and you can plant separately and they'll form into new plants. So here's what it looks like at the end of the season. So if these detach and break off, you can still form a new plant from that. But again, they're going to be genetic clones of that original bulb that they came from. But bulbs are under the ground, whereas something called a bulbil is going to be above ground. These are tiny compared to a regular bulb. They're bulb-like organs though. As you can see, they do look kind of like an onion or a garlic. Okay? And these are produced on aerial stalks. They're held above the ground. So you'll find that a lot of our wild garlics and our wild onions around here are more prone to reproduce this way than they are sexually. Every once in a while you'll see the flowers that happen later in the summer. We're not going to get a chance to see the flowers. But you may see these. And these did not form from an egg and a sperm fusing together. These are asexual. They are genetic clones to the plant that they're on. If they break off and fall off, they'll get a new plant that will be genetically identical to the original plant. So bulbils, above ground on stalks. The bulbs are actually underground. Then we have our clonal fragmentation, which is where you've got a piece of the plant that breaks off and roots to form a new plant. Like our little different pads here, these are what they call digits on prickly pear cactus. These will break off occasionally, and then they'll start to form new little adventitious roots and they'll form into a new plant. Okay? And then here we've got something called Mother of Thousands. If you were in my plant ecology class, you got a chance to see this one in person. It forms new little plantlets along the margins of the leaves that have little roots that stick out from them, and they'll just fall off and form into new little plants. But again, these plants didn't form with the fusion of egg and sperm. Right? They are asexual propagules. They're genetic clones of that original parent. And there's other types as well. Some plants can actually form seeds right, without the process of colonization or fertilization occurring. They're exact clones of the parent, and yet they are making a seed. They can do parthenogenesis. So if you're familiar with that term with animals, it can happen in plants as well. So there's lots of different ways this can happen. But this complicates that biological species concept definition because we don't have the capabilities of having interbreeding here if the plant is going to reproduce this way primarily. We'll wait till it decides if it wants to advance. Let's try this. There we go. All right. So what can we use instead? Because there are other ways to define species. A lot of people like the morphological species concept because all you have to do is look at the appearance of the organism and look for differences. As long as they look different from each other under that definition, they would be considered different species. The problem with that with plants is a lot of plants look the same. A lot of different species look identical to each other. It's really hard to tell them apart based on the morphological characteristics. And if you look around our classroom right here, you run into a problem too if there's variation within the species. Because none of us look the same. And by that definition, we would all be different species. And we're not. So these you don't have to memorize, nor do you have to write them down. I just want to give you an idea of some of the other things that people have used and some of the ones that are now becoming more favorable because they work a little bit better for some of the species today. So you have the one we just talked about, biological species concept, which is based on reproductive isolation. You have recognition, which means that the two organisms have common fertilization systems. You have the phonetic one, where the smallest groups of individuals that are distinguishable from all others are considered a species. Evolutionary, also sometimes referred to as the phylogenetic one, a lineage of populations that has had a common evolutionary fate through time. Essentially, they're on the same branch within a cladogram. Autoapomorphy, species that are defined by their possession of at least one shared derived trait. By now, we should know what a shared derived trait is. This is something that you get when you do cladistics and get a specific type of phylogenetic tree called a cladogram that shows you where those new evolutionary traits appeared in evolutionary history. And if you have one on the position in the phylogenetic tree, everything above that should have that trait unless they lost it later on. So that would be their defining feature that could potentially define them as a species. And then we have genealogical. Individuals of a species are more closely related to one another than any individuals outside the group. Lots of different options, right? But the point of this is that it's complicated. Defining a species is complicated. And there are some professors in our department that don't like defining species at all, hate species definitions. Dr. Pitts is one of them. And he just says they're useless because there's no real way to do it for all species that exist. Right? So you'll see that species are defined in different ways sometimes with plants. Okay? And you can use a lot of different types of data to figure it out. So plant systematics defines species using a wide array of evidence. It sometimes uses morphological data, the appearance, the anatomy, the structure of the plant. But sometimes, and what we're starting to see now, is you use molecular data. We're starting now to utilize this more because the techniques that we needed to be able to look at the DNA and RNA are more refined now and less expensive than it used to be. And we're better at it. This used to be something that was reserved primarily just for the R1 institutions that had a lot of money. And to a certain extent, sometimes it still is, depends on how sophisticated technique you want to use. But more and more people are getting to the point where we can start using this method for defining species. So, what we're going to talk about next, usually not the favorite chapter of the students because a lot of us have not seen this material before. We're very briefly going to discuss a little bit about molecular systematics. Okay? So we're going to start talking about DNA and DNA techniques. We may have discussed some of them in 140, but if you have only had 140 as your only cell molecular class, you may not have had any of this other stuff. So we're going to kind of take a very, very, very shallow view of it. In the sense, we're not going to delve too deeply into it, but I want you to know some of this because this is the direction taxonomy is going. And that applies more than just to plants. This applies to animals as well. If I could go back in time, I would take a lot more molecular classes than I did so that I could do more of this with my work. Okay. So what exactly is molecular systematics? Well, recall the definition for systematics, just the general definition for systematics. You are studying the diversity of life. That means not just living things today, but extinct things as well. You're looking at all of these different species and you're trying to figure out how they're related to each other in terms of their evolutionary history. We're trying to figure out phylogenies, which again is not something we can ever truly know because we've never been there for the entire lifespan of a species. Okay, so we can only hypothesize how species are related to each other based on the data that we have. And this is one of the types of data that we have. So molecular systematics is using DNA and RNA for relationships among the organisms. So we're specifically using DNA and RNA to make these phylogenetic trees, hypothesizing how species are related to each other based purely on their DNA. There's some big advantages to doing it this way, as opposed to using morphological characteristics, anatomy and structure and appearance of organisms. There's a lot of advantages to going directly down to the DNA and looking at that instead of the physical features of the organism. Okay, first reason, you get a lot of data. Think about how long a DNA strand is. Right? This is minuscule compared to what the length of this would be. We're talking millions of base pairs long. That's a lot of data. Each one of these positions right here is something that you can compare from one species to another. So for example, we have one species here, we have another here, and here. We've just taken a little segment of their DNA. We've lined them up and we're going through and we're looking for differences and similarities. That's what we do when we try and figure out how species are related to each other. We're looking for similarities and differences. The more similarities you have, the assumption is the more related you are to each other. So think about how long this is. That's a lot of comparisons that we can make across the DNA strand. If you're looking at an organism and its physical features, there's a limit to how many things you can compare. You can compare their size, you can compare their color, you can even look at the individual lengths of the bones, but there's only so many characteristics that you can compare. There's a lot more of the DNA characters that you can compare than the physical features, so more data. Second, your interpretation is going to be much easier looking at the DNA than it is to be looking at physical features. So we've got two birds right here, very, very similar looking. Same species, different species. Right? There might be some subtle differences in maybe the depth of where this black streak goes right here, maybe a little bit difference in the banding pattern on this. Physically though, they don't look that different. It could just be individual variation like we see within our populations in terms of the physical features. They could be the same species. That means using something like the morphological species concept wouldn't work, right? And somebody in the class may say, I think they're different enough to be different species. And somebody else in the same class may say, I don't think they are. I think it's minuscule, I think it's negligible, and they're the same species. That means it's subjective. If different people are going to have different opinions about what's different enough for two species to be considered different species, that's what I mean by subjective. But go back up here to our DNA strands. Everybody know what these letters stand for? What is the G? Guanine, cytosine, thymine, adenine, right? And in RNA there'd be uracil, right? Right? Those are our nitrogenous bases, right? that help make up our DNA strands. Is a thymine different than an adenine? Yes, right? No ifs, ands, or buts, right? They are definitely different. It's not, well, maybe it's different, maybe it's not. No, they're different, right? Definitely different from each other. That's not something we can necessarily do with physical features. You lose some of that subjectivity. So we're a little more confident in our interpretations that yes, in fact, these are different. Okay. Number 3, we can compare distantly related species that don't really look similar to each other. Okay, so say for example, we've got these 3 plant species right here. We're trying to figure out how they fit in our phylogenetic tree, and we're trying to group together the ones that are more closely related to each other. And we're trying to figure out this one in the center, whether it's more closely related to this one on the left or the one on the right. They don't really look a whole lot alike at all. what are the similarities we see here? None of them have the same shape to their flowers. None of them are the same color. They don't have the same clustering pattern to their flowers. We find the leaves look very, very different from each other. So how do we know which one this one's more closely related to, if there's really no physical features that they share? Well, if you go down to the DNA, you might find those similarities. And what we find is that this one is actually more closely related to the one on the left, the wisteria, than it is over here to the snowdrop. Now the reverse works as well. Okay? In this case, we're trying to compare very distantly related species that don't look alike at all. But you can also compare very closely related species that look exactly alike. It actually doesn't matter how much they look alike, because if they are truly different species, you're going to find those differences at the DNA level, even if physically you can't see it. So even though their characteristics may be the same, you go to the DNA, you'll find the differences. Okay, now here's one of the biggest problems that you run across when you try to compare the physical features of organisms and use that for your basis of comparisons for phylogenetic trees. Your environment has an effect on your appearance. You could have two individuals that are the different sizes, even though they're genetically the same, because they got different amounts of food as they were growing up. Right? So one may have been able to develop a little bit faster, a little bit bigger than the other one. Otherwise, they're exactly the same, even though their size is different. Here's another good example. We've got hydrangeas here that are genetically all the same. And you have some of them that have these bluish colored flowers. We'll get into more specifics later what those flowers really are. And you've got some that have these pinkish colored flowers here. They're exactly the same. Genetically, they're exactly the same. They are, in fact, the same species. The only difference between them is the pH of the soil. So the pH of the soil in this case, whether it's acidic versus basic, can change the availability of aluminum to the plants. If the plant is able to get the aluminum, right, based on the pH of the soil, it'll be pink. If it doesn't get it, it's going to be blue. So all that's different is just their environment that they're in. Otherwise, they're the same plant, the same species. You can mess with your grandmother with this. Start changing the color of her flowers, and we see what happens with that. But that doesn't happen on the DNA level, right? Your phenotype is affected by your environment, but not your genotype, not your genetics, not your DNA. It's your physical features that are impacted by your environment, not the actual DNA. And so going down to the molecular data factors out this environmental effect. You find out that these are the same species. In this case, they're clones. They're exactly the same. Now, there's some other advantages to this too, because it used to be that people primarily used morphological data to make their phylogenetic trees. So they make these cladograms and get these phylogenetic trees and say, here's all the data I have based on anatomy, structure, and here's how I think they're related. Well, now that we can look at the DNA, we're starting to do the same thing with the DNA, and sometimes what we're finding is the two trees match each other. Super awesome, because remember, a phylogenetic tree is a hypothesis. So if you have two different types of data, morphological data and DNA, molecular data, that's giving you the same cladogram, the same phylogenetic tree, you can be more confident that this is probably the phylogeny, the true phylogeny for this group of organisms. You can never prove it, but the more data you have to support that, the better. The more confident you are in your conclusions. Okay? So oftentimes it supports the groupings, the trees that we find based on morphological grounds, their physical features. Doesn't always happen. And actually in your book, you're gonna find some groups of organisms we're gonna talk about are gonna have multiple cladograms that are all based on different types of data, and none of them match, okay? And we'll get into that a little bit later too. Also, sometimes this allows systematists to choose between competing hypotheses for certain phylogeny. So let's say that two different groups of scientists have been working on a group of organisms to try and figure out what their phylogeny is, what their phylogenetic tree should be, and they both come up with different trees. Now you have this other group of scientists that's gone to the DNA and is using molecular data to make a tree or construct a tree for this group of organisms, and is trying to see which of these two hypotheses based on morphological data is right or supported. So the one that has the most evidence for it is probably the more likely scenario. And number 3, there have been some groups of organisms, I'll use turtles and tortoises as an example, I'll use the conifers as an example, that we just cannot figure how they're supposed to fit into that tree. Sometimes you'll see dotted lines in phylogenetic trees because they're like, well, I know it's around here somewhere, they're supposed to go in here, but I have no idea how this branch is supposed to look. And that's very frequent. But that sometimes happens if you have only one type of data to work with. But if you now add in molecular data and you do the same analyses with that, you can sometimes figure out where the placement of the tax is supposed to be that were problematic. It can help you with that bit. So hopefully we're getting a new appreciation for the molecular data, even if we don't personally work with it, and understand the value of working with this type of data. So now we have to figure out how to do that. Now we're specifically going to target plants in this case, because this is a botany class. So let's talk a little bit about plant genome. Let's talk about plant DNA, types of DNA that you're going to find in plants. Plant cells have 3 different DNA components. They have chloroplast DNA. This is DNA that's going to be in an organelles, or organelles inside the cells themselves. They're going to have mitochondrial DNA, different organelle, that's in plant cells. And yes, plants do have mitochondria, just like we do. We don't have chloroplasts, but we do have mitochondria. Because both humans, or animals, and plants do cellular respiration, aerobic respiration. That's how we break our sugars down and get our ATP. We both do it, so we both need mitochondria. And mitochondria have their own DNA. But then you have the cell's DNA, the actual cell's DNA that's in the nucleus. So we call that nuclear DNA. Now, generally speaking, your chloroplast DNA and mitochondrial DNA in plants is usually inherited from one parent. Usually it's the mother. Here's why. Okay? The egg is bigger than the sperm, okay? Which means that you could potentially fit organelles like mitochondria and chloroplasts in that egg. So when it fuses with the sperm, yes, you're getting the nuclear DNA from the dad, but you may not be getting anything else besides that. Okay, so you may have only your mother's, this is plant, chloroplast or mitochondrial DNA, not the father's. There are some exceptions to that though. New studies have actually shown in gymnosperms, they get their chloroplast DNA from the father instead of the mother. So it does depend, but usually it's one parent that you're getting it from. Nuclear DNA, on the other hand, you get from both parents. Essentially, the offspring are a combination of the 2 parents, the mother and the father. Now, these types of DNA, very different in size. The nuclear DNA is by far the largest, because these are the genes and the DNA, the instructions that the organism needs for it to be able to build itself and function, all its life activities. This is the main bulk of the DNA and the chromosomes. You can see right here the size is kind of a size range depending on species, is how big it can be, 1.1 times 10 to the 6th or 110 times 10 to the 9th kilobase pairs. So that means take these numbers right here and multiply by 1,000, and that's how many base pairs you have in these DNA sequences. It's a lot of DNA. Mitochondrial DNA, on the other hand, will only be about 200 to 2,500 kilobase pairs, and chloroplast DNA is the smallest, about 135 to 160 kilobase pairs. Yes?
Well, we need to know the actual sizes.
Not the actual sizes, but who's largest, who's smallest, who's middle, yes. But that's just, you can get a feel for how much bigger the nuclear DNA is than the chloroplast mitochondria, significantly bigger. They're also different in terms of their shape. And when I say shape, I mean if you took the DNA or the chromosome, you actually stretched it out, right? Some of them are gonna form into a ring, they're gonna be circular, and some of them are gonna form into a long line. So mitochondria and chloroplasts have circular chromosomes. It's a ring. We're gonna talk about why later. It's because these two types of organelles are thought to, at one point in time, to have been bacteria that were engulfed by an ancestral plant cell. Bacteria, prokaryotes, have circular chromosomes. And so therefore, chloroplasts and mitochondria have circular chromosomes. And it's their own DNA. It's unique from the cell's DNA. Another line of evidence that it came from a bacteria. And we'll come into that later with the endosymbiosis theory in the next chapter. Nuclear chromosomes, though, are linear. So you stretch those out, they're gonna be lined. They're just like ours. You take the DNA from our nucleus, our chromosomes, stretch it out, it's gonna be a long line. Now, some of these types of DNA are gonna be a little more useful for classification and making cladograms and phylogenetic trees than others. One of the things that you want the DNA to be is you want it to be pretty consistent, constant. If it mutates all the time, if it rearranges a lot, that complicates things because you're trying to, for example, take DNA from this individual and from this individual and compare them. If it's constantly gonna be rearranging, it's not gonna give you a good idea of a good comparison between these two species as to whether they're related or not. And some of these types of DNA do more rearranging than others. For example, the mitochondrial DNA rearranges frequently. So yes, you could use it, right, to make your phylogenetic tree, but it's not as useful for making phylogenetic trees because you may get a different result the next day, right, or the next year, even if you're looking at the same individuals. So not very useful for relationships. The other two, though, a bit more stable. So frequently we use chloroplast DNA and we use nuclear DNA for our comparisons and our phylogenies.
So what's the point of rearranging the DNA? Like,
It's not necessarily just trying to do that. The organism is not making a conscious decision to rearrange its DNA. It oftentimes happens in the process of replication, natural processes in the mitochondria or the cells themselves. Think about mutations, they're always random. Our enzymes are good, but they do make mistakes. Okay, so step number one, if you're gonna use molecular DNA to make a phylogenetic tree, the first thing you need to know is you need to know something called the sequence of the DNA. So you remember that picture I showed you earlier where it had the 3 different taxa and we had all those letters A, G, T, 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 that sort of thing? That's what we're trying to figure out, okay? We need to figure out the precise order of our nucleotides. These are the nucleotides. What order are these in? That's what's important. And how does that order relate or compare to another species? Easier said than done, right? Because DNA is microscopic. It's not something where you just go down there and read and say, okay, this is an adenine, this is a thymine, guanine. This is not as easy as it sounds in terms of its process. But before we can even do this, understand that many of the processes that actually determine the sequence of DNA will destroy the DNA in the process of doing that procedure. Right? So the first step is you've got to make sure you've got enough DNA left over that you can run multiple tests on this. So you may not always get a lot of DNA though, especially if you're talking about a fossil. If you're looking at a fossilized species, you may have just a little bit of DNA that's available and not a lot. So we've got to make some copies of that. Make sure we got enough copies to work with and we've got some left over if we wanted to do more procedures in the future. So we use a special process to do this. You've probably heard about this in some of the other classes. We're going to get into some more specifics about how it actually works this time. It's called PCR, And that stands for polymerase chain reaction. Polymerase is dealing with an enzyme that we're gonna use in the process. Know what those initials stand for, polymerase chain reaction. Know also that this process does not determine the sequence of the DNA. All it does is copy it. Right? All we're doing right now is we're just copying the DNA. We're getting enough of a sample for us to work with, then to ultimately do the procedure that's going to determine the sequence. PCR just copies it though. And you may not be copying the whole genome. You may be just copying a fragment or a piece of the DNA that you're interested in looking at. Because oftentimes we're not comparing the whole sequence of DNA from one species to another. We're comparing sections, related genes, as an example. So how does this process work? Let's walk ourselves through the steps here. First thing you gotta do for the PCR process is you gotta combine everything you need to do this together into one little vial, or one little tube. Here are the things that you need. You need your original DNA. This is the DNA that you're interested in copying, right? The one that you want more of. Put that in the vial, right? That's step number one. Second, you're gonna put in something called primers, which I'll define in a little bit as to what primers are. Then you're gonna put in an enzyme, an important one. This is gonna do the work of actually making the copies. It's gonna be some sort of a DNA polymerase. But specifically, usually for the PCR process, one of the main ones that they tend to use is something called Taq polymerase, and I am going to explain why we use that. We got an enzyme. And you want to put a whole bunch of free nucleotides in there. So loose adenines, guanines, thymines, cytosines, just free nucleotides. Throw them in there, as many as you can, right? Because you're going to need those to build your DNA strands, your copies. Step number two, you're gonna heat this solution. Now in nature, this is not how it works, right? This is not how the plant copies its own DNA. And it doesn't use heat in the process. Let me explain why. We have to do heat because what we have to do is we have to unzip our DNA. DNA is double-stranded. Everybody remember that? Right? Got two sides to the ladder. You need to pull that apart, right? So that you have two single-stranded pieces right here that are going to serve as templates for this copying process. Now in nature, the way the plant normally does this when it copies its DNA is it uses an enzyme to do that. We can't do that in the lab. So instead what we do is we heat it. And when you heat things like enzymes, when you heat things like proteins, when you heat DNA, it denaturizes. And that's what that means. Right? So we're going to unzip it. All right, I'm going to stop here for today. We're going to get back into this a little